Hello everyone, this is Ma'am TK once again, and welcome back to Clinical Bacteriology Laboratory. So, we're down to the second part of our lesson on the correct morphological classification using different staining methods for bacteria identification. Now, before we really dig into this part of the lesson, let us have first a quick recap on what we have discussed the previous weeks. No, so do you remember what we did? What we have tackled on? So we tackled on the bacterial shapes, the structures, their arrangements. We also tackled on the different bacteriological structures that just a certain type of bacteria possess, yung mga unique structures that would help you further identify a bacteria, and also the procedures by which you would need to master in order for us to really demonstrate these bacteria and their structures. Okay? Now, welcome to the second part of this lesson, and that is on simple to selective staining. Now, the vast majority kasi guys of individual bacterial cells, no, they are nearly completely transparent or colorless. Okay, that's why the visualization of microorganisms or bacteria in the living state is most difficult. That's why I reiterated yung previous topic na if the bacteria is not dead yet during the preparation of the bacterial smear, then the staining procedure will definitely kill the bacteria. Now, you would be wondering naman, bakit nyo ako sinasabihan na hindi colored sila? Yung pinakita nyo sa amin last time, colored yun. Scam ka ba, ma'am? Of course not. Ginawa natin yung colorful. That was visually enhanced. Guys, ginawa natin yung may animation that is to spark interest in you. But of course, they would not look like that under the microscope. Except of course, dun sa mga pictures na pinakita ko that I really stated that these are the bacteria and how this is how they would look like under the microscope. So yung mga ganun na pictures, syempre yun talaga itsura nila. But apart from those pictures, the other things, they were just enhanced, okay, for you to better to really appreciate the structure and the bacteria themselves. But don't worry, even though the bacteria that we will be working with, they are colorless in nature, we have the stains and the staining procedures that would help us further examine these bacterial structures and entirety of the bacteria. It will be presented now. Okay, so now before we really dig into this topic, here are some topic highlights to guide us along the way. So for today's topic, we'll be talking about the staining itself. Also, we'll be talking about the nature of the dyes and the procedures, the meat of this topic. Okay, on the different procedures that we will have to master para mas maklaro at saka mas ma-test natin, mas ma-identify natin yung ating bacteria. So, let us start. So, what really are stains, guys? Okay, your stains, otherwise known as dyes. Okay, these two terms can be used interchangeably. These are organic or aqueous preparations that impart a variety of colors to tissue or microorganisms. Now, this further states that staining allows the bacteriologist to further enhance the visibility of the microscopic bacteria. Kasi guys, in order for us to really study the structures, okay, and to differentiate the bacteria into specific groups for diagnostic purposes, we have developed and we have developed biological stains and also the staining procedures in conjunction with light microscopy in order for us to fully equipped with the major tools in microbiology. Now kasi guys, hindi enough na may stain tayo. Alright? Apart from choosing the right stain, you would also need to consider what is your goal in staining. Now, not all stains are created equal kasi guys. Do you remember histopath? Of course, kasi syempre fresh pa yung rattle, no? Yung ating struggle sa histopath. So, do you remember the topic on stains? Okay, with those things in mind, you can now picture out how bacterial stains function as well. Alright? The stain that you would need to use will vary on a lot of depende sa mga factors involved, okay? So, it could be on what you want to determine. Do you want to know how they look like, what are their shape or arrangement? Or, further, do you want to see the different types of structures to really identify whether or not these bacteria possess the structures that would make them unique, or to make it a more complex na goal, what do you want the staining procedure to accomplish? Do you want to differentiate the different types of bacteria found in the sample? So depende kung ano yung goal mo. Okay? Kung ano yung gusto mong mangyari, gusto mong makita, doon tayo magbabase sa kung anong stain ang gagamitin. Okay. Now, your stains, apart from the fact that there's a lot of them, they vary not only in color, they vary not only in purpose, but most importantly then, they vary in composition. Okay, that brings us to the nature of dyes. Now, your dyes or your stains, they contain substances that are made up of ions. Now, do you remember in your chemistry wherein you talked about ions, positive and the negative ions, diba? If positive and negative ions come together, they form what substance? They form salts. Okay, this goes to show that your dyes, which are composed of various ions, they are actually composed in its majority of salts. Okay, your dyes have salts. Now, your ions, guys, they function differently. Okay? Depende sa kung ano yung nature or depende sa kung ano ang type ng dye mo. Okay? The ions assume different roles in different dyes. Okay? This will be made clear to you now. If ang ion, okay, if the ion contains the coloring material, or this is the ion that contains the color, ito yung magbibigay ng kulay sa bacteria or sa bacterial structure, we call that ion as the chromophore. Okay, again, the ion, it could be the positive ion or the negative ion. Okay? Depende kung positive ion or negative ion. Depende sa kung sino sa kanila ang may kulay. Okay? That would make it now called the chromophore. Anong mangyayari doon sa other ion, ma'am, na walang kulay? Hindi yung ion na hindi magbibigay ng kulay, hindi magbibigay ng, or walang pulos? No, of course not. Yung ion na hindi chromophore, ma'am. What do you call that ion? We call that counter ion. Okay. To further explain this, if your chromophore is the positive ion, meaning yung chromophore mo ngayon, yung kulay mo ngayon nasa positive ion, okay, yung positive ion ang magbibigay ng kulay sa bacteria or bacterial structure, we call this specific type of dye as a basic stain. Okay? Basic stain if it's in the positive ion, if the chromophore is the positive ion. Now, if the negative ion is the one that gives the color or is the one that has the color, yun, or the chromophore in the stain, we call this particular stain as the acidic stain. Okay, why is this important, ma'am? By the way, guys, I'll give examples of basic stains. Yung mga ang chromophore kasi yung positive ion. We call that, yung examples, you have your crystal violet, your methylene blue, your safranin, malachite green, carbolfuchsin. Yung sa acidic stain nyo naman, we have your eosin, your picric acid, nigrosin India ink. Guys, there are a lot of stains under basic or acidic stain. Now, please lang guys, just a quick segue. Now that you have better control over your time, kasi nasa bahay lang naman tayo, make it a point to not just rely on what we give you or what your professors give you. I'm not talking not just on bacteriology but on third year as a whole or like medtech as a whole. Okay. yung mga PDFs, lectures, what and whatnots, yung mga binibigay ng mga professors. Please do not just rely on them. Also, better yet, supplement them. Alright? Back them up with your own readings. Grab your books, guys. This is the best time to practice and master, no, the skill or art of reading. Now, I tell you guys, because if reading comes as easily or as natural to you as breathing, then definitely you will finish this course with flying colors. No? Mas colorful pa sa stains na pag-uusapan natin. Corny mo, ma'am, no? Sige na. Sige, ma'am, sige. Sige, saba, ma'am. Oy, move on na tayo. This is the best. Basta guys, ha? This is the best edge that you could ever build and sharpen for yourselves. Alright? Reading. I tell you, your future self will thank you for this. So read, alright? Apart from the things that we give you. So going back, now that you know about the basic stains and acidic stains, how they, what are the ions that make them or that give them function, alright? Ano yung sa mga ions na nandoon yung may kulay. We move on now to the basic charge of your bacteria. Your bacteria kasi guys, the cell wall, it is negatively charged at pH of 7. Okay, yan yung kanyang charge because of the fact that the cell wall is made up of proteins like peptidoglycan, your teichoic acid. Basically, these are components of the cell wall that would render, okay, the bacteria, yung kanyang outer covering, as negatively charged. Now, based sa stains and the ions that they contain, okay, the colored ions that they contain, what do you think will stain the bacteria now kung negatively charged siya? Syempre, it is the ion that has the negative, that has the opposite charge, and that is your positive ions, right? So, what are your positive ions na chromophore? Ano yung stain na yun? You have your basic stain. So, basically, your basic stains now are the ones responsible for coloring, for imparting stains on the bacteria. Okay. So what happens to the acidic stain naman? So anong gamit niya? Bakit siya nag-i-exist? Okay? Kaya wala naman siyang, di naman niya pwede na i-color yung bacteria. Given that the bacteria is negatively charged, it will repel. Of course, your acidic stains, they are manufactured still for a purpose. What happens now? Your acidic stain will perform its function not on the bacteria wherein it will repel. It will be repelled with negatively charged ions on the bacterial structure or the cell wall. Rather, your acidic stain, if not the bacteria, it will stain the background. Okay, it will stain the background. Okay, this will make more sense later. Okay, basta alalahanin nyo yung particular na ano na yan, ha? Particular na concept. Huwag nilang kalimutan. Okay. Now that we're done with the nature of dyes, let's move on to the staining procedures. Alright. Alright. Your staining procedures come in two forms. Again, highlight ko lang, ha? The determination of the shape, the sizes, the arrangement of the bacteria, and even the structures that they contain, they are all useful in the initial steps in identifying an organism. And much more so, okay, their function is further enhanced by identifying the staining capability or the staining characteristic of a certain bacteria or their structures. Now, the staining procedures are either direct or indirect. What do you mean by direct, ma'am? The direct type of staining procedure is when it directly stains the bacteria and the bacterial structure, or the organism to be studied absorbs the stain, making the cell or the structures, they appear with color. So, ang kinukulayan niya again, yung bacteria. Directly, yung bacteria. Your direct staining procedure are further subdivided into different types. You have your simple staining. Now, from the name itself, simple. Kasi, dito sa type ng staining na to, you only make use of one type of dye to study the organisms. Okay? Walang special structure na makikita, walang ibang ma-highlight. Kasi, again, what happens here, the stain evenly dyes the entire bacteria. Ganun siya kasimple na type ng procedure. Okay. A much more complicated one is your selective or your special staining. Do you remember the structures that I've presented to you? Yung mga bacteria structures, diba? Okay. Those structures, guys, they cannot really be examined when you use simple staining. Okay, if you want to demonstrate those specific structures, you would need to go into selective or special staining, because it is in here that you can isolate the specific parts or the structures of the organism for you to further study them. And then last, on the direct staining procedure, we have your differential staining. This is a higher order na type ng staining. Why? Because not only can you see the bacteria, not only can you possibly look at the structures, but rather you can also differentiate different bacterial types between another. Okay, kasi syempre yung ating samples naman, hindi lang naman yun, if ever bacteria are present there, no, hindi lang naman isang bacteria ang nandyan. So you have your differential staining to further differentiate them amongst their peers. And they have a lot of staining procedures for that. You have gram stain and acid fast stain. Don't worry. Okay, for this topic lang, we'll focus on the simple, selective, and special. The differential staining, it will come sa next na topic after this. Okay, para hindi masyado tayo mabombard. Okay, for your selective and special staining or differential staining, guys, no, diba I told you these are much more complex compared to simple staining wherein isa lang talaga yung stain na gagamitin natin. That's why it's important din for us to know kung ano yung mga different reagents na makikita dito. Okay, disclaimer, just a really important note. Okay, the reagents here that are used in the selective and differential staining, ito yung format. Okay, ito yung format na ifa-follow natin. However, these reagents can still vary sa specificity. Okay? Depende kung ano yung specific staining procedure na gagamitin natin, na ifa-follow natin. But, almost always, your selective or special staining procedure or differential staining procedure will have these reagents. Okay? Mag-vary lang sila sa kung anong type ng reagent ang gagamitin. Okay. Your selective or special and differential staining always has an initial or primary stain. Ano ito, ma'am? Okay, from the name itself, initial or primary. Ibig sabihin, this is the first stain that you will use in the procedure. Okay, ito yung unang stain na mag-i-impart ng color. Okay, anong stain, ma'am? Depende. Again, depende siya kung anong procedure mo. It can be methylene blue, malachite green, depende. Okay, depende sa procedure. But almost always, may initial or primary stain ka. And then, we have your mordant. Okay, what is a mordant? Yung stains nyo kasi, yes, it can give color to the bacterial cell wall, the structures, okay? However, kailangan mo ng specific na reagent that would allow these stains to stick to the bacteria. Now, your mordant, it is that substance that will form a bridge between the cell and the initial stain para mas magdikit yung stain mo. Your mordant comes further into different types. We have your physical mordant. Now, in the physical mordant, you don't really need to use a chemical here, or hindi mo kailangan gumamit ng kung anong chemical. Kasi what you do here in physical mordant, you subject the smear into either heat or cold. Okay? Walang chemical na gagamitin dito. Heat or cold lang. Physical mordant. Okay? Para mas mag-stick yung initial stains sa bacteria. If you have physical mordant, syempre meron din tayong chemical mordant. Now, in chemical mordant, from the name itself, we make use of chemicals. Example, iodine, ferrous sulfate, tannic acid. Again, depende sa procedure na ipo-follow nyo. Your mordant is closely followed by your decolorizer. Okay, decolorizer, what does it do? It decolorizes, it removes the color. Now, you would like ask me na, ma'am, bakit parang ba't palang wrong? Kinulayan mo siya, nag-effort ka pa na padikitin yung kulay sa cell using a mordant, tapos tatanggalan mo ng stain? Of course, this is actually a very crucial step in your special staining and differential staining. Why? because it removes the initial stain or the stain from those bacteria or structures na hindi mo naman kailangang i-test or hindi mo naman kailangang examine. What happens here is, the bacteria in question, yung bacteria na importante sa'yo na para matest mo, makita mo, sila na lang yung may kulay once it undergoes decolorization. So, yung mga unnecessary na mga substances doon sa smear, or yung mga bacteria who do not have a specific structure or composition in them to grasp more tightly sa initial or primary stain, sila yung mga na-decolorize. That will help you further differentiate the bacteria and the bacterial structures found in the smear. Sige lang, don't worry. I know this is quite tricky pa. You will know more about this as we go on. Okay? So that's the function of the decolorizer. Okay, that will help you further differentiate. And of course, we do not end there. Alright? The last equally important na reagent in these types of staining, yung selective and special order differential, we have your counter stain or secondary stain. Okay, what happens here, yung mga na-decolorize na mga bacteria doon sa after decolorization, syempre bibigyan natin sila ulit ng kulay through the use of the secondary stain. Okay, ma'am, paano yung ma'am? Masasapawan? No, hindi necessary. Okay? Hindi masasapawan necessarily yung mga hindi na decolorize kanina. Okay? Again, this is just to further differentiate the bacteria present in your smear amongst each other. You will know more about this later, alright? So now that we're done with the direct staining procedure, of course, we also have its counterpart, and that is your indirect. Okay. If your direct staining procedure directly stains the bacteria and the structures, where your indirect, it's the complete opposite. In indirect staining procedure, we do not stain the bacteria, rather, we stain the background. Okay. Let's start first with our direct staining. So, starting with the simple staining. Again, what happens here is that we only make use of one type of stain. So, the stain is flooded across the smear, and then there's no special structure that is distinguished. Example ng stain that is employed for simple staining is your methylene blue. So, what happens? You just flood it with stain, you wash it, alright? After flooding it with 1 minute, tapos i-wash mo siya with water, you blot it dry, allow it to dry, and then look at it under the microscope, and that's it. You don't need to do anything, just one dye. That's simple staining. That's how simple staining is. Now, let's move on to a much more complex type of staining, and that is your selective or your special stain. Okay? And since we have different structures, we also have different types of stain. We'll focus on the endospore stain, capsular stain, granular stain, and flagellar stain, respectively. So, let's start first with your endospore stain. Your endospore stain, okay, may specific name tayo dyan, this procedure, and that is your Schaefer-Fulton stain or Schaefer-Fulton method. Now, just quick lang na review. What are spores again? Okay, spores, these are bacterial structures that are tough or dormant, and they are non-reproductive. Ito yung mga hibernating parts ng bacteria mo. Okay, they are usually present if the bacteria is subjected to harsh environmental conditions. Okay, you differentiate your endospores, okay, or cells from cells with endospores with those bacterial cells that are called vegetative, or ito yung mga vegetative cells, simply these are cells na walang endospore. So yun lang yun. Vegetative cells, sila yung mga nagre-reproduce. So you can do the differentiation using this endospore stain. And again, your endospore, okay, being very tough, it is not easily colorized, guys. Hindi siya agad nakaka-pick up ng stain. And kung na-stain man siya, it's also not easily decolorized. Okay? Because of the presence of apart from yung dipicolinic acid, calcium, and peptidoglycan complex, it is also further coated with a keratin layer. Kaya grabe niya impenetrable. Okay. So further about that, let's move on with the procedure. Now, what do you do in Schaefer-Fulton method, ma'am? First, you prepare a bacterial smear. Remember how you prepare a bacterial smear? Go back to our previous procedure or previous topic, no? So after you've already prepared your bacterial smear, napadry mo na siya, you start with the staining. Okay, what you do is you flood the smear with the primary stain. What is the primary stain for Schaefer-Fulton, ma'am? That is your malachite green. Okay, your primary stain. Now, what you do after putting the malachite green, you heat the flooded smear to gentle steaming, okay, for 5 minutes. Okay, usually, you make use of alcohol lamp or nilalagay siya sa slide warmer. Basta makita mo na parang may steam na maproproduce, okay, you do that for 5 minutes, okay. You do not allow the smear with stain to dry up. Make sure na may timer kayo as you do this procedure. Now, once you're done, you pour off the excess stain, yung excess malachite green, and rinse it thoroughly with tap water. Okay. Now, Ma'am, nasaan dito yung mordant? Ma'am, nasaan yung dito, yung decolorizer? The mordant is the physical mordant that you did, yung steaming. Okay, that is the mordant there, the heat. Your decolorizer here is your tap water. Okay, so now that we have decolorized it, we flood the smear again with the secondary or yung counter stain natin. What is our counter stain for Schaefer-Fulton, ma'am? That is your safranin. Okay, we do this for 1 minute. And then, we rinse thoroughly with tap water, deblot dry, and then we examine it under the oil immersion objective. Now, as you can see dyan sa right side, that is an actual microscopic na na view. So, nakita nyo, the malachite green, the primary stain, stains your endospore green or blue-green or something. And then your safranin now stains those that are not the endospores, which are your vegetative cells. So, you're not able to differentiate now. Okay? Mas na-identify nyo ngayon kung asin yung endospore, di ba? So, apart from Schaefer-Fulton stain, meron din tayong other stain for endospore. And I want you to read on that, guys, ha? We have the molar stain. But ang usually used for endospore is your Schaefer-Fulton. Okay, remember this. Now, we move on to the second na structure, and that is your capsule. You remember your capsule, guys? So, your capsule, this is the structure that is a protective outer covering of the bacteria that would protect it from phagocytosis, yung immune system defense mechanism. Now, fun fact like, the presence of the capsule increases the pathogenicity of the bacteria. So, para mas makapaghasik ng lagim ng iyong bacteria, okay? Kasi mas defensive siya, kaya meron kasi siyang capsule around him. And in identification of the bacteria, guys, again, the presence of the capsule will help in determining the proper medical intervention, okay, to combat this pathogenic feature, yung capsule, and eventually to kill the bacteria that causes the disease in question. Okay. And like your endospores, your capsular staining do not absorb most times. In fact, mas nakaka-absorb yung endospore. Your capsule not really. Okay? So, the most routinely used na procedure for your capsular stain is your dry India ink or nigrosin stain. Guys, this is actually an example of an indirect staining. Okay? What happens here, ma'am? So, what we do is we suspend the organism. We take a bacterial sample, okay, in a drop of 6% glucose solution placed on a glass slide. Now, what is the glucose solution for? Actually, guys, the presence of glucose, okay, does a lot to the bacteria. Ang daming nagiging effect niyan sa bacteria, yung metabolism niya, okay, so basically processes in the bacteria. Let's just put this in a more basic perspective, ha. The presence of the 6% glucose would help the bacteria further demonstrate, okay, the presence of capsule. So, pag may glucose, mas nagkakaroon or mas nakaklaro yung capsule ng bacteria if ever the bacteria is capsular in nature or may capsule talaga siya. So, yun. After putting a 6% glucose solution, we place an equal volume of the first stain, or actually this is the only stain, the India ink and nigrosin. And then once we do that, the India ink or nigrosin rather, we mix them properly, and then we perform the blood smear. Okay, what is the blood smear technique, ma'am? It's a type of smearing technique wherein we put two slides together. Ang isa nasa 45-degree angle, and then we let it slide across the other. We let it slide across the other slide. Okay, I hope this makes sense. Sige lang, you will actually, ang blood smear technique, it is introduce sa'yo sa inyo sa HEMA. That's how we perform how we perform the hematologic smears. I don't know if you've seen it, yung nagpakuha kayo ng dugo, yung parang may ginilagay na isang drop ng blood sa smear, tapos yun yung na-perform yun minsan ng mga medtech sa harap nyo. Yun yung tawag sa blood smear technique. Anyway, you will know about that in HEMA. So that is what we're going to do in your dry India ink or nigrosin. Now, once you've performed the smearing technique, you allow the smear to air dry, and then you do not heat fix. Ma'am, bakit? Because heating, okay, will cause your capsules to shrink. Alright? They will shrink so much na hindi nyo na sila makikita, so useless. So, sige, so ang gagawin nyo lang, you just allow it to air dry. Alright? And then, you cover the smear with another stain, and that is your safranin. Alright, and after covering the smear with safranin for 10 seconds, we wash off the excess stain, we rinse it with tap water, and then we air dry, and then, like all bacterial samples, we examine it under the oil immersion objective. And that is what's gonna be seen under the microscope, guys. Ang nangyayari, your bacteria is not stained. Rather, the stain colors the background black. Okay, it colors the background black, and it outlines the outer layer, or the outline the outline of the capsule. So it will look like this. This is what we call now as halo effect. Okay, so para siyang halo ng angel dyan. So yun yung nangyayari. That is your dry India ink or nigrosin stain. And of course, yung capsular stains mo, it's not only limited to that. We also have another type of staining procedure for your capsule, and that is Anthony's. So please read on that, alright? Okay, next is our third structure, and that is our granules. So remember this particular picture, diba? It was presented to you last topic. So, ang tawag sa granular stain natin, guys, is Albert stain. So, what Albert stain or the Albert method? What happens here is we just prepare a bacterial smear, like how we do it sa mga previous na smearing procedure. Then, we stain it with what? We stain it po with Loeffler's methylene blue for 3 to 5 minutes. Okay, and after 3 to 5 minutes, we rinse it thoroughly with tap water, we allow it to air dry, and then finally, we examine it under the OIO. Now, after this, guys, this is how you're gonna see your structure or your granules. They would appear brownish black, okay, yung granules, while the cytoplasm and even the background would appear green or even up to light blue. Okay, that's why your Loeffler's methylene blue is otherwise known as a metachromatic stain. Kasi it produces a metachromatic effect doon sa ating bacterial smear. Now, last on the list, we have your flagellar stain. Okay, your flagellar stains, guys, are there to really help us examine or makita talaga yung mga flagella, because in reality, guys, your flagella are so thin. Okay, these are hair-like structures that help the bacteria or propel the bacteria forward. Okay, these can be long, di ba sabi ko, sometimes they would appear longer than the actual bacteria itself. However, they are very, very thin that you cannot really see them sometimes. So, your flagellar stain helps us to better see them. So, ang nangyayari dito sa flagellar stain, bale, the method pala of flagellar stain is known as your Leafson stain. So, what we do is we wash a growth from a bacterial culture. Usually, ang bacteria dito na ginagamit, syempre yung bacteria that is known to have yung flagella. Example, Proteus vulgaris. Okay? Sige lang, you will know more about Proteus vulgaris as we go along. So what we do is we wash the growth of Proteus vulgaris or any bacteria with proposed or parang thought of as merong flagella with 3 ml of sterile distilled water, and we gently agitate it. Then, we let it stand for 7 minutes. Ma'am, what is the function of letting it stand? Bakit dito sa flagella stain lang nagpa-stand ng suspension, ma'am? Suspension pala yung tawag doon, guys, ha? Yung the bacterial sample plus the distilled water. Ang tawag doon bacterial suspension. Bakit, ma'am? Because by letting it stand, yung bacterial suspension, we let the flagella to unravel, okay, to really regenerate and to really extend. Okay, so parang magkalat talaga siya para makita talaga natin siya once they are stained. Okay. Now, then while waiting for the 7 minutes, you can do other steps of the procedure, and one of which is making a ring of 1 to 2 centimeter in diameter on a glass slide. So you can use wax there, depende. Basta you make a ring on the glass slide, and then yung bacterial suspension na pina-stand you for 7 minutes, you get a drop and you run it across the ringed surface of the slide, okay, doon sa loob ng ringed surface. You air dry, but again, you do not heat fix. Hindi mo siya i-heat fix. Bakit, ma'am? Kasi po, by fixing, usually, di ba, we use, what mordant do we use to fix? Di ba, heat? Heat now can cause a damage to your flagella. It will disintegrate your flagella, ha? It will destroy it. Flagella is too fragile, guys. Okay? So, after siya ma-air dry, you cover the ringed area with the stain, your leaf sun stain. And then, you let it stand for 10 minutes. You wash it with your distilled water, allow it to blot dry, and then you examine it under oil immersion lens. And that is how it will look like under the microscope. Also, guys, your flagella can also be demonstrated not by another procedure. It's not with a stain, but rather this technique. I think you're familiar with this. We've discussed this a previous topic, and that is your hanging drop technique. Because again, your flagellar stain. This can be seen under procedures that test for motility then. Okay, so one example for flagellar stain, or rather a procedure to demonstrate flagella, is your hanging drop technique. And then lastly, we go on to our indirect staining procedure. Your negative or relief staining. Guys, actually na-mention na natin to kanina doon sa ating capsular. And that is your dry in the ink or nigresin staining. So the background here is stain instead for contrast. So may halo effect tayo. Okay, and that is usually used for capsules. Because capsules, again, do not really take up stains. So, instead na mahirapan kayo, you just stain the background. Alright. So, that's it for the first part of your staining. So, we've tackled on simple staining, on selective and special staining, the different types of staining procedures, your direct and indirect. Okay, the nature of the dyes and what staining is really all about. Now, prepare yourselves for the demonstrations that would come after this, and of course, for the last part of the staining next week, and that is your gram staining and AFB to be given to you by Sir Kuday. Okay, thank you so much for listening, guys. See you again soon.